Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Difference between actin and myosin filaments. Table of contents. Actin definition. Myosin definition. Key differences. Actin versus myosin. Examples of actin. Examples of myosin. Actin definition. Actin is a group of globular proteins widely found in eukaryotic cells, playing a crucial role in providing shape, structure, and mobility. It is present in all eukaryotic organisms except for nematode sperm. Actin proteins are highly conserved and engage in more protein-protein interactions than any other known protein. Unlike other proteins, actin can transition between two states: monomeric, G-actin, and filamentous, F-actin. This transition is regulated by nucleotide hydrolysis, ions, and various actin-binding proteins. Actin serves as the monomeric unit for two distinct cytoskeletal filaments, microfilaments, and the contractile apparatus. It plays essential roles in cell division, motility, and signaling. The dense network of microfilaments allows cells to remodel in response to environmental stimuli, such as increasing adhesion to form tissues. Vertebrates express three main actin isoforms. Three alpha isoforms are found in skeletal, cardiac, and smooth muscles. Beta and gamma isoforms present in both muscle and non-muscle cells. In muscles, actin filaments are short, measuring between 2 to 2.6 micrometer in length and approximately 0.05 micrometer in diameter. Actin filaments in muscles are separated by actin-binding proteins like alpha-actinin, which binds two actin filaments while maintaining space for myosin. Alpha-actinin is a key component of the contractile apparatus in muscles. Each actin filament in a muscle fiber consists of two intertwined strands of actin protein. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Myosin definition. Myosin is a superfamily of motor proteins that, along with actin, plays a fundamental role in muscle fiber contraction. It is classified as a motor protein because it functions as an enzyme that converts chemical energy into mechanical energy. Myosin acts as an ATPase, moving along actin filaments by coupling ATP hydrolysis with conformational changes. All myosins consist of one or two heavy chains and multiple light chains. Genomic studies have identified 13 different myosin types. Each myosin molecule shares a highly conserved structural organization, including a head domain, which serves as an actin-activated ATPase responsible for generating movement, a neck domain, associated with several regulatory light chain subunits, an effector tail domain, which varies among myosin types and determines their specific cellular functions. Myosin filaments are larger than actin filaments, measuring approximately 4 to 5 micrometers in length and 0.01 micrometers in diameter. In muscle cells, myosin primarily exists as myosin II, a polymeric molecule with a long, rod-like tail domain that assembles into thick bipolar filaments. The dimeric head domain of myosin II alone is capable of generating movement in the presence of ATP. Beyond muscle contraction, myosins also perform various functions depending on the specific myosin type and the organism. Despite variations across species, the structure and function of myosin remain highly conserved, with mammalian myosin being compatible with the actin of unicellular organisms. Key differences: actin versus myosin. Actin and myosin are two essential proteins responsible for muscle contraction and movement. Let's explore their key differences. Definition. Actin is a globular protein found in eukaryotic cells that contributes to cell shape, structure, and movement. Myosin is a family of motor proteins that interact with actin to enable muscle contraction. Location in the sarcomere. Actin is found in both A and I bands. Myosin is restricted to A bands. Size and structure. Actin is shorter, measuring around 2 to 2.6 micrometers, and thinner, with a diameter of 0.05 micrometers. Myosin is longer, about 4 to 5 micrometers, and thicker, with a diameter of 0.01 micrometers. Nature and molecular weight. Actin is a globular protein with a lower molecular weight. Myosin is a motor protein with a higher molecular weight. Abundance and surface texture. Actin is more abundant in muscle cells and has a smooth surface. Myosin is less abundant and has a rough surface. Filament composition and cross bridges. Actin filaments contain actin, tropomyosin, and troponin, and they do not have cross bridges. Myosin filaments are composed of myosin and myromyosin, and they form cross bridges. ATP association and filament ends. Actin is not directly associated with ATP. One end is free, while the other is attached to the Z-line. Myosin is directly linked to ATP. Both ends are free, and its head domain interacts with ATP. Role in muscle contraction. During contraction, actin filaments slide into the H zone. Myosin does not slide, but generates force by binding ATP and interacting with actin. General location. Actin is found in muscle fibers, microfilaments, cell membranes, and cell walls. Myosin is primarily found in muscle cells, playing a crucial role in contraction. In summary, actin and myosin work together to produce movement, with actin providing the structural framework and myosin generating the force needed for contraction. Their coordinated interaction is what enables muscles to function effectively. Examples of actin. Microfilaments. Microfilaments, also known as actin filaments, are protein structures that contribute to the cytoskeleton of a cell. These filaments are composed of actin protein polymers that interact with various cellular proteins. Each microfilament has a diameter of approximately 7 nanometers and consists of 2 intertwined strands of actin. Microfilaments serve multiple functions, including cytokinesis, cell shape modulation, and cell motility. Additionally, actin structures located near the plasma membrane adapt based on the specific function of the cell, such as forming specialized structures in the middle piece of mammalian sperm. Examples of myosin. Myosin in stereocilia. Myosin 3 proteins are located at the tips of stereocilia in the inner ear. Stereocilia, which are structurally similar to the microvilli of the intestinal tract, measure approximately 10 to 15 micrometer in length. These specialized mechanosensing organelles detect fluid motion in the ear, playing a crucial role in hearing and balance. Myosin 3 forms a complex with PDZ domain, containing proteins, working alongside actin to facilitate the response of stereocilia to sound waves. By enabling movement of the cilia, myosin helps stereocilia adapt to changes in sound waves and fluid motion. Additionally, genes encoding myosin proteins have been identified in other regions of the ear, such as the cochlea. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!